Dear friends, good morning. Wish you all current 75th Azadi Ka Mahotsav and forthcoming festivals in the month. It is my proud privilege to welcome you on behalf of Professor Brahm Singh Horticulture Foundation, BSHF, a not-for-profit organization, and my own behalf to this fifth H webinar series entitled Future Indian Horticulture. BSHF is thankful to NIPA, New India Publishing Agency, New Delhi, for sponsoring this webinar series. I am happy to welcome co-organizers Dr. Preetam Kalia, ICR Rafi Ahmed Kidwai Awardee, former Head Division of Vegetable Science and Coordinator, School of Horticultural Sciences, ICR IRI New Delhi, and Dr. Shailendra Rajan, former Director, ICR Central Institute of Subtropical Horticulture, Lucknow. As per the title of the series, the emphasis in this series of webinars is on future horticulture. And as you know, future horticulture must be science and technology driven. Hence, we would see in coming decade new vegetable varieties or hybrids with useful new traits introgressed using modern biotechnological and IT tools, particularly omics and data science. Omic technologies fetch the information from genome, transcriptome, proteome, metabolome of the vegetable plants and link them with the phenome of the crops to understand the physiological, biological, molecular mechanism at the level of gene expression, regulation, translation, and metabolic pathway. The generation and interpretation of the omic data have taken a long way to modulate other crops and vegetables too. Techniques of the science of omics are useful in improvement, disease and pest resistance, and tolerance to abiotic stresses in vegetable crops. The next decade would see vegetable improvement using biotechnology tools on which Dr. Tushar Kanti Behra, Director ICR Indian Institute of Vegetable Research, would enrich our knowledge along with other crop improvement technologies and tools. Friends, questions can be raised in chat box, which we would take up after the talk. Now I request co-organizer Dr. Preetam Kalia ji to formally introduce Dr. T.K. Behra.
Thank you, sir. Good morning, everybody. Most respected. Are you getting me?
Yes, sir.
Am I audible? Most respected Padam Shri Professor Brahma Singh ji, founder and chairman of BSHF, and co-organizer Dr. Shailendra Rajan ji, and today's speaker none else than Dr. Tushar Kanti Behra, who would be talking on vegetable improvement in next decade. Dr. Behra doesn't need much introduction, as he is well known in the vegetable world, in the country and abroad, who hails from Brahmpur, Balasore in Odisha, has had his graduation from OUT Bhubaneswar and master's and doctorate from Indian Agriculture Research Institute, New Delhi. He earned his postdoctorate fellowship program at University of Wisconsin, United States of America. Presently, Dr. Behera is Director of Indian Institute of Vegetable Research. Prior to taking over as Director of Indian Institute of Vegetable Research at Varanasi last year, Dr. Behera served as right from scientist to principal scientist at IERI in the Division of Vegetable Science. He also became professor of discipline of vegetable science, and he had been nodal officer of ICR IERI Jharkhand Hazaribagh. Dr. Behera has contributed significantly in improvement of most of the vegetable crops, but in specific, he has developed 23 varieties and hybrids in brinjal, cucumber, and gourds, which were most of them were released by CVRC and also SVRC and notified by Government of India. Many of these varieties has been licensed by the private seed companies. He has also been instrumental in isolating gynoecious lines in bitter gourd from its field form Momordica carentia variety muricata and introgressed recessive gynoecious gene GY1 into commercial cultivars of bitter gourd, which were also licensed by the private seed companies through MOU. Dr. Behera introgressed gynoecious trait F. locus in Indian cucumber also using marker-assisted backcross breeding. He has been instrumental in developing a variety for protected cultivation called as Pusa Rasdar. And Dr. Behera has guided 12 PhD and 2 MSc students at IERI. He served as chairman of subcommittee to develop a postgraduate course curriculum on horticulture for IM ACRE program of Myanmar and ANASTU of Afghanistan. He has contributed more than 125 peer-reviewed research papers, edited 3 books, and also contributed 15 book chapters, 7 training manuals. Dr. Behera has had traveled to USA, Australia, France, Russia, Thailand, Libya for academic assignments and was member of 11th 5-year plan for horticulture, plantation crops and organic farming. He has been a member of RSE of Mushroom Institute at Solan and Bihar Agriculture University, Bhagalpur. Dr. Behera has been on institute management committees of various institutions, and he has also served as the FAO International Consultant in vegetable breeding at Libya. Dr. Behera is a fellow of many organizations. Important to mention are Fulbright, Nehru Academic and Professional Excellence. He is also fellow of National Academy of Agriculture Sciences, Indian Society of Plant Breeding and Genetics, Indian Academy of Horticultural Sciences, and Indian Society of Vegetable Science. Dr. Behera has been conferred with several awards, and the ones worth mentioning are 20th Hari Krishna Shastri Memorial Award, Best Teacher Award for Higher Education, Dr. Biswajit Chaudhary Memorial Award, Dr. Kirti Singh Gold Medal, Fulbright Nehru Academy and Professional Excellence Fellowship, Hooker Award, DBT Associateship, Dwarkanath Memorial Medal. Presently, Dr. Behera is associated with as Secretary of Indian Society of Vegetable Science, and he is member of Executive Council of Indian Academy of Horticulture Science. He also served as editor of Horticulture Society of India, Agriculture Research Journal, and Indian Society of Agriculture Sciences. Besides, he has been assistant editor of CGCR, United States of America, and also he served as the treasurer of Horticulture Society of India. So, with a lot of contributions of Dr. Behra to the vegetable improvement program and the uh, self attainment that he has obtained because of his contribution to the system, he has been selected to rule the roost to take forward the vegetable improvement and general vegetable science program at Indian Institute of Vegetable Research as director. So, with these few words, I invite Dr. Behra to take the floor and deliver the talk for the benefit of the viewers. Dr. Behra, please.
Thank you, sir, for your kind words. And I think I am audible, sir.
Yeah.
Yeah. So, good morning to all of you, and most respected Dr. Brahm Singh, sir, and Dr. Pritam Kalia, one of the mentors for my own programs at IRI. Dr. Rajan, we also have a colleague, senior colleagues to our horticulture SMD. My dear friends and the all, I can see that many people are joined and for this particular webinar. This is the work what we have to do in the next decades, that vegetable crop improvement programs of our nation as well. And in that time, I just to brief some of the things like what we did it in the last 5 decades, because we are observing the Azadi Ka Amrit Mahotsav. In that case, we have a lot of works already been done in the last 50 years of our research in vegetables, particularly the improvement aspect. The improvement means it also related to the productions and the crop protections as a whole. So I think we have a tremendous increment in the respect to, but still we have many to work upon, because in the improvement programs we are very much of are not concentrating to the vegetable as a crop improvement program like other cereal and other crops in India as well. But if we see in the other country, the vegetables, particularly most of the vegetables, in case of your tomato solanensis vegetables, you can go for cucurbitaceous vegetables. You can see that vegetables improvement already been done with respect to this, using the different molecular tools or the biotechnological tools, which is also very important for our program. Being in the crop improvement program, we need to have a lot of infrastructures with respect to this vegetable crop improvement program. But if you see, India as a whole, very few people working in the different crops of vegetables. We have about 120, 25 vegetables are grown in India, but we are working only 20 major vegetables and maybe 20 more minor vegetables for the improvement program. And if you see the vegetable crop improvement, we are generally from the beginning don't have so much of infrastructure facility. Also, we don't have so much of worker training to these particular technological issues, which is very, very important, and that required to go for a very precise way of crop improvement program in vegetables. We have a human resource development program is going on throughout the country, but it is not so ample enough to go for the vegetable crop improvement, which you need to concentrate in the next decade. These are very, very important things. So here in this program, I just in the topic, I'll go at that what we did it for the last 50 years of our program. Then we'll again the next decades, or what I have to go for the next decade. There's some of the issues I will flag on, and that we have to as a whole, and the workers and the vegetable crop improvement program, whether it is a crop production scientist, or whether it is a crop improvement scientist, or is a plant protection scientist, we have to, or in the private sector organizations, we have to work together for the improvement of vegetable crop in this country. So that is the important thing. So I will go for this one. And the first, as I mentioned, that what we have in 5 decades of increase in the improvement program. If you see the major infrastructural boost in the country, we have started the regional station at vegetable programs at Cochrane in Kulu in 1949. Then it is again as a vegetable breeding station in Cochrane, it's in 1956. Then in 1967, we have Indian Institute of Horticulture Research in Bangalore as established to take the activities of all horticultural crops, including vegetables. Now, if you see this creation of the horticulture as a whole, then there's a horticulture department is in there in Indian Agriculture Research Institute, New Delhi. And after that, we have in 1971, there's a ECRIP programs started in India to go for the multi-location trial or testing of the different vegetables of this country. Again, we have in the development of 1986, we have project director in vegetable as it at IRI. During 1992, it is shifted to Varanasi, where now I am posted. After 1992, there is a tremendous increase in the vegetable research is concerned for the country, either in the through of ECRIP programs or the institute programs. And most of the ICR, as well as the state agriculture universities, scientists, they're working on this particular aspect of vegetable crop improvement program. And 1999, we have Indian Institute of Vegetable Research programs has been initiated, where we have 20 major vegetables and 22 underutilized vegetables we are working upon. I think this is the boost of vegetable productions, not only the public sectors, the public-private sectors also has a lot of contributions with respect to vegetable productions of this country is concerned. Now, if you see in 1951, where we have only 16.5 million tons of vegetables out of 2.84 million hectares, now we are jumping about 193. Now, as you mentioned in 2020, 21, third estimate, 22, third estimate was about 200.45 million tons of vegetables. As you consider that in the as ICM are concerned, we have to at least 350 grams of vegetables will be there. But we, as a gross productions of concern is concerned, then we have already achieved that one. That is, we are in the gross per capita availability is about 400 grams per person per day. But if you see the post-harvest technology, post-harvest losses and other losses is concerned, and we are about 300 grams of vegetables each will be there. So, as you see that the growth is tremendous. We have per capita availability is about 3.3 fold increase. Production is about 11.2 fold increase. And we have productivity also 3.1 fold increase is there in this particular sector. If you see the global trend in a vegetable as concerned, it contributes about 14.8% of total vegetables areas of this country and 14.3% of total production. And we are the second largest producer of vegetable. There is a phenomenal increase, as I mentioned, that we have so many increase in the lot of crops. We are doing, as I mentioned, 125 vegetables we are working together. But if you see the achievements, we have a vegetables throughout the country, specific vegetables for a specific region, which is very, very important, not that of other crops. We have a choice of local preferences of vegetables with respect to different region is concerned. If you see the ECRIP trials we have, and we have generated about 573 varieties and developed and notified of 30 vegetables in the last 5 decades. Also, if you see the open pollinator variety 340 around, and we have 178 or 180 hybrids are developed. Now you are also concentrating on the development of varieties resistance to different biotic stresses. That is about 57. And we developed also the technology, which is very, very important. Without technology, we cannot see the harvest, the potentiality of the different crops or the variety as a whole. And you see that we have 450 production technology, 213 seed productions technology, and 53 physiology and biochemistry work. And also we have 165 disease and 135 insect pest management technology, particularly IPM, also been developed with interest to different vegetable crops. If you see the world Indian positions in the global vegetable production, if you say that we are some of the vegetables definitely we are first or second, but also some of the vegetables we are 13 or 14 or 22nd or 25th. So as you go along, definitely we have to increase. That is the things or the challenge that how we can also have to productions in the country as a whole. We can also, as a world as a whole, we can produce is the first rank in India, which is the major changes to be done in these next decades, particularly for vegetable crop improvement program. If you see that the average potential and productivity of the different vegetables is very, very important. As you mentioned, we are already threefold increase in the productivity is concerned from the last 50, 70, 75 years. But if you see that the average production is tomato crops and any other crops, eggplant or okra or peas, you can see we are far, far below than the world average. Also, not this world average, maybe maximum productivity country as a whole. Tomato, if you see that we are having 24.34 million tons per hectares, but in US you can see the 98 average production should be there. So in this table we can see that how much we have to increase to go for the productivity level by which we can also see that the maximum productions can be achieved for a limited area of our products. As you know, we have these areas are limiting. We have a very small areas for cultivation of vegetables, but vegetables are very important because it also give to the livelihood security of these people, also to the nutritional securities of this country. Now, if we have to consider the small farmland hunting, vegetable is the only option for the farmers or the stakeholders to go for this type of vegetable productions, as well as it can also give you a food. You can also give you the economic importance of this particular family. Also, it also using as a nutritional security of the family, which is very, very important. Now, many of the programs are coming out. The UN programs is also associated here in Varanasi region. They have started some of the work on the Poshan Suraksha vision with respect to India as a partner of that one. And we are also trying that to have the school village programs where you can also have these nutritional programs we can start in the schools itself. If you see the demand or the productions of the vegetables in the country, in the very few states like Uttar Pradesh, West Bengal, Bihar, Odisha, Madhya Pradesh, Gujarat, and Maharashtra, they are producing most of these vegetable crops of this country. And if you see the crop improvement program as a whole, you can see, you can see that in the country as in the world itself, when this breeding program started, it is not the breeding program which is started and the other crops, but it started in our vegetables crop, that is in pea, in 1866, where laws of genetics, Mendel, as you know that there are a lot of Mendel principles, the three Mendel principles are there, which is used for the crop improvement program. Then we have hybrid breeding in the commercial hybrids in the world in 1920s. Then 1928 we have first reporting mutation breeding in the maize. So we have the Green Revolution program started, and which is also seen in the 1960s. Now coming to the different programs, as Dr. Ramsingh has already mentioned, we are in the different way of omics region, like genomics we have started, metabolomics we have started, proteomics we have started, and many things have started. And the world is always in the advance. And other than India, they have started the work. And many of our scientists from this country also involved in the other country to develop this type of technology to go for the crop improvement program as a whole. But if you see the genomic studies started in case of your cucumber beets crops or the vegetable crops in 2009, when the first that in cucumber they have the sequence, the full genome sequence of this cucumber started in 2009. Then after that, tomato and other crops already been initiated by the programs of different countries, and most of these genomic selections have been started by other countries as a whole. But now I will say that some of the programs like genetic resources, we have these active germplasms, which are basically working by the different state agriculture universities as well as the ICAR Institute. And these are the programs where we have the active germplasms in the year 2006 to 7, how much we have this active germplasm. Now we have increased in 2022. Some of the germplasms we have increased. We have characterized the germplasm. We also have in the germplasm bank or the gene bank in the NBBG. If you see this genetic stocks is concerned, these are the past breeding materials. Through these our evaluations, we can find it. Many of the genetic stocks also been registered at NBPGR. If you get seedless pointed gourd, you have gynoecious lines, you have tropical kale, you can have a CMS-based hybrid systems in case of radish. You have a tomato resistance to different leaf curl viruses, as well as you have root and tomato resistance lines. Then gene pyramided lines also will be available in our gene bank, which can be utilized for the further breeding programs as a whole. If you see that some genotype which is very, very novel traits, like if you have sponge gourd, which is having the basmati rice flavor, high frequency mel in case of summer. So these are some of the examples I'm putting. I think many of the state agriculture universities and the ICAR Institute, as well as the private partners, they have a lot of genotypes or the novel with the novel traits. And most of these cases, they're using that for the breeding program, either through the quality improvement program or in the program of the hybrid development program. They have a specific trait-specific breeding they are doing. And I think that will also coming to the next generations or next decades, we can have a lot of crop improvement programs or a lot of quality traits programs also to become up in the future itself. If you see, these are some of the examples I'm putting. Resistance to variety resistance, which is through the AICRP, which has all been released. And some of the private also, they have contributed tremendously with respect to the resistance to different pests and diseases in this particular crop. Now we are talking about the biofortified nutrient-rich vegetable varieties. As you know that Dr. Kalia is working in this carrot. He has a lot of vegetable variety, carrot, and the cauliflower itself is working in this aspect, particularly to develop the new lines and the varieties. But some of the examples also I can put it here, that in case of our institute also trying to have some of the orange cauliflower or the different color segments of the of radish and carrots is concerned. These are the very important, because nowadays we are talking about these nutrient-rich varieties and advanced lines in this particular crop, particularly in case of watermelon, in case of muskmelon, in case of cauliflower, as well as in case of carrot, which is very, very important for the development of the nutrient-rich varieties, and where we can have a different segments we can grow. Also, we can have a tropical carrot type where you can have this type of pigments, which is very much of nutrient-rich and can be also associated with the programs. But most of the cases we are doing only this hybridizations or selection process, or we have to go for developing the new lines in that particular aspect. Some of the cases we have also tried with the marker stud breeding, development of OR gene related genes in case of cauliflower, particularly which is rich in the beta carotene content in case of cauliflower. So these are some of the things we have tried, and most of the still we are in the different backgrounds. You have to put this type of color pigments or in this type of nutritions, so that it can also be more easily visible or accessible to the farmers or the consumers itself. Because as you see that the transgenic approach which did it in the golden rice, that is for this particular for iron rich and other things. But we have in the nature, we in vegetables, we have very, very good lines of the crops are available, particularly for the underutilized vegetables, the leafy vegetables as concerned. We have a lot of vegetables where we have the iron rich type of things are there, and we have different other color pigments, which is essential for the antioxidant properties also be there in these particular crops. So these things we can also have in our research programs. I think the next year we can do this one. We started with this gene pyramiding, and that is most of the cases, particularly in tomato, which is a success story for gene pyramiding with respect to multiple disease resistance in tomato. Many of these our breeders in the country, they use the material from AVRDC, whether it's the private or the public sectors. Some of the things we have taken or we have procured from USA with respect to different resistance gene in tomato. Also, they have taken it from the different species, which is very, very important. The related species, which are mostly utilized, like Solanum pilulifolium, Solanum hepatitis, and many of these species also utilized for developing this type of resistance in the different crops. Now we have a pyramided lines also available, whether it is in private sector, whether in other public sectors. We have a different multiple resistance varieties like root and nematode resistance, lead blight nematode resistance, and also TLCB. Now we started some of the work in case of early blight in case of tomato, and we also seen the genes associated with this early blight and the markers associated with the early blight, which are we are using for the further improvement or pyramidal lines in our own varieties is concerned. If you see the story of that particular, we have our Karakshak, and it is very much now in the northeastern region, northeast region as well. In the south, it has a very, very potentiality for the production. But in some of the cases, because of the viruses and different type of viruses in different other regions, we have to definitely interrogate some other genes with respect to resistance to these particular leaf curl viruses in case of tomato, as well as also the IHR also developed some of the Orca samrat or Karakshak, bacterial resistance, olive blight, and tomato leaf curl viruses. And we have also worked with the Cassia Oman and Cassia Viman with respect to this type of resistance program in our program. So many of these private companies, they have their very good hybrids, which is also having resistance to this one. Most also, we now are trying to have this resistance in our own materials with the bacterial, particularly abiotic resistance, that is a drought and the high moisture stress conditions. We have to also build some of the varieties, the hybrids, and that is also underway in the process to develop. Now, if you come into this, significant achievements in the molecular breeding in vegetable crops, and if you see the cucumber is one of the model crops, particularly for this one, there is a lot of improvement done from 2009 onwards after the genome sequencing of cucumber, which is, I just give you an example. In tomato, there a lot of work has been initiated. In brinjal, a lot of work has been done. So these are the things where they are major crops. We are also have a lot of work. But as I mentioned this cucumber, because many of these, as it is a model crop, that is very important. If you see this as an example, for the sake I'm putting here, the phenotypes of repetitive mutants in the case of cucumber, you have a tremendous type of genes are available with respect to the different phenotyping of this particular crop. If you see the leaf type, you have glabrous, you have round leaf, you have super compact leaf, you have short hypocotyl, yellow plant type. So these are the leaf characters. The genes are already been find out, and it is already been observed that the association of these particular traits, how much we are concentrating on this just to go for trait-based breeding in case of cucumber. If you see the variations in the spine size, fruit place, maybe white, orange, yellow, green, these are also different mutant types also available in this cucumber. Being origin, the cucumber center here. In China, that is mostly we have these two species like Cucumis haruki and another is Cucumis hystrix. So we cannot forget the Cucumis hystrix, which is also near border to the China. We have these materials also there. And now this development of the Cucumis hitibus, again, which is resistant to different stress or the biotic and abiotic stresses, now giving to a lot of improvement in case of cucumber. So this is, I am putting some of the examples that how we have genes, we are trying to have the mutants, the phenotypic markers, everything are available in a particular crop. So that sort of things we have to initiate in the different vegetable crops to have a trait-specific breeding program and to have an increase in the productivity and quality of the vegetables. These are the important things we have to do it. If you see this genome sequence, you can say that you can count some of these crops in case of your vegetables. If you see only cucumber or melons or some of the crops we know, but tomato we know. But if you see the worldwide, is a genome sequence crops in horticulture crops, there are several crops has been genome sequence has been done, and out of which already 42 has been done. So I just see in the paper that already it is now to 68 genome sequence done in case of vegetable crops. So there's a tremendous the genomic platforms are used by the different other countries, but India is concerned, we are very lack with respect to this genome sequence is concerned, the vegetables and the other horticulture crops is concerned. But we have to work. If you see 90 to 95% of the genome sequence is done by the Chinese groups. So this is the very important that how much we have to go and how much you have to go for these facilities to create infrastructure facility for study the modern science by which we can improve our vegetable crops. This is very important. That's why I put it here. If you see the sequence started from 2009, and we have in 2022 many crops including Momordica, Cucurbita, and other crops already been sequenced. So we have a tremendous potentiality to have this type of genomic platforms where you can use our programs for the program. If you see, there's some of the examples I put it in cucumber, because how the size of these fruits can also be regulated by the few genes, like carpel number genes, which is very, as you call it, a clavata gene, or that is which is associated with the WS protein, particularly for the pseudapical meristem basis. We can also find out, or floral meristem basis, we can find out, or basis of the carpel number, we can, the genes associated with this one, we can find out that how we can improve, or how we can manipulate the size of the sepals, petals, stamens, and carpels. So this is very important. By identifying this type of genes, we can have also both the linkage way, and also in the study with different other way association mapping programs. We started that work in case of US. We are part of this program, and we have seen that these are the way we have to go for identify different genes. And in case of tomato, also the same thing has been happened. By this different gene, we can also find out the shape, size, and also the clustering behavior. All the genes are available, and based on that particular study or the gene association study, we can see this one. So by the way of your molecular markers, you can identify which are these having carpel number 3, which are having carpel number 5. So that also can be, you can identify this cell, by which you can also see that what shape it will be your crop is there, what shape your fruits will be there. So these studies also have a basic strategy. I think we have to concentrate that way in our future breeding program, so that by manipulating our own marker state breeding program, we can develop the newer lines with a different size, different color, different type of crops of vegetables we can also generate ourselves. So this is the thing I have shown it here. The expressions of the genes also we studied with respect to the different carpel number, and also with respect to the different examining with the microscopic examinations by the meristem size. We can also find out that these are some of the lines of the genes, which is lines which is having the different carpel number over there. If you see this chromosome locations of the different genes is concerned in cucumber, we have a different clone genes. We have also fine map genes are there, which is I also put it here in the red and black color. So these are some of the genes which is very important, and we identify already in case of cucumber. The locations of the different flower, vegetable organ, flower, fruit, and so on, based on the QTL programs by the different scientists, they throughout the country, they can find it here. It's an important part here, the chromosome 1, chromosome 5, and chromosome 6 is very important in case of cucumber, where a lot of genes respect to the fruit type, fruit color, everything will trace color, flower color, everything is there with respect to these 3 chromosomes. So these are also been informed properly by the different scientists. And also you can see that the disease resistance genes and the QTLs in the different, as well as in the chromosome 1, 2, 3, and these also having different downy mildew, powdery mildew, fusarible resistance, gummy stem blight, all the genes can be located, and the different chromosomes, we can also map them properly. Now, if you see the thousands of genes have been identified with respect to cucumber, we have respect to the different genes locus, and we identify the biparental mapping system. Now, there's a very good process is the association mapping. We are also the GWAS, by which number of genes are identified for the different, specific to the different traits is concerned. We have our small scale studies also can be done with respect to SSR marker. Also, we have studied the SNPs, which is having very important nowadays, particular precise genomics selection, which can be also done through this particular way of different improvement in the infrastructure. The programs also initiated in the transgenic, as you know, in case of the earlier in case of brinjal, we have a Cry1C genes, which is already been there in case of particular for the fruit and shoot resistance in case of brinjal. But that again, we have a transgenic cell, but we have now the sort of ethical issues and some other issues. Still, it is not in the process in the finalization to the coming to the farmers as well as to the conjugate cell. But we have to also see the transgenic, by which we have to definitely, in other countries they have already have a given a program of transgenic, like in case of Bangladesh we have already this transgenic, but still we have to again do and see the work which we have to go for a long way for the transgenic. The transgenic with respect to the gene pyramiding in the tomato, like abiotic stresses, we have drought, heat, salt, and particular for TDRB gene and BCJAR gene, which is already been also transits all been there, and we have also created some of the populations of the generations we have already advanced the generations with respect to drought and heat and salt and cold resistant genes in this particular way of transgenic approaches. But still, we have a long way to go in this way. As you mentioned about this crop improvement program, I will not go for many of the things, but as you show all these varieties, all the hybrids, whatever we have also to develop the vegetable production technology, which is also very important. It should go side by side. It's not that only you can go for the breeding programs or the crop improvement program, but the crop improvement program also associated with the vegetable production technology. Because if you are not giving the proper way of technology is concerned, the vegetables requirement of the input by the vegetables, it will not express, the genes will not express without the production technology or without the precision production technology what we have. You can see the climate changes are already there in our in the world itself, and we are having a different expressions of the genes with respect to the production technology, the nutrient efficiency, the water. If you have a different type of waters, biotic and abiotic stresses, definitely the expressions of the genes also is there. So we have also done with respect to the tremendous increase in the production technology with respect to high technology nursery, where we have a nursery regime, particularly if you have seedlings are very growth or they are very much of good vigorous, then only it expresses the fruiting habit. So that's why you have to have some nursery when we have already some initiatives or the people have already in the worldwide they have done it, that this is the nursery technology we have to put it. Similarly, in case of micro fertigation system, where you can case of fertigation system is very, very important in the world. We are now going to have a less input or less use of the labor, and by the mechanization process we can also have this system by which the fertigation work can also be done. We have to also see that how much water we are wasting for that. So that we have to also have some of this mechanism by which we can also reduces the use of water or the irrigation facilities to these particular crops. So these are the things are improvement already been done. I will not go details of this one, but as you know that some of the technology which is really proven very good way to go for, particularly in case of mulching, whether it's organic mulching or the plastic mulching, but we have a lot of concern with respect to this water use efficiency with respect to this particular crop is concerned. So in vegetables, as you know, these are very huge, they use huge amount of water as well as also the nutrients. So in that case, we have to also see the genotypes, which is also less requirement of the inputs, and that also we have to see, and how best we can use these inputs, particularly for to have a potential yield of this particular vegetable variety is concerned. As I mentioned, mulching is very important, whether you go for plastic mulching. But again, if you go for plastic mulching, we have to again see that environmental pollution. Again, we have to see that one. If you see that environmental pollution in the plastic is very, very now going along. If you see in some other countries, it's very, very restriction is there. But in our country, the mulching through this plastic mulch, we are not taking care of. The growers, the farmers, the research institute also, they work on this particular aspect of plastic mulch, that they put it in the plastic mulch field itself because it is not a degraded material. So we have to see that some of the works we have to see that some degraded plastic mulch can also be used, or how best we can do it for our mulching program. So that also we have to see. It is some simple example of the crop where we can have the simple staking of tomato. You can have 70 to 75 tons per hectare. But still, we are not practiced because of this input. The farmers cannot pay more amount, particular for staking in this one. But the simple techniques, we can also increase our yield. This is very, very important that in case of seed productions, in case of bottle gourd, or the crop productions in case of bottle gourd in the power system. So these are, I just mentioned it here to have a look that some of the small intervention also can have a better improvement programs in this crop. If you see the protected cultivation, if you see in the worldwide, you can see the protected cultivation is grown throughout the country. But if you Indian concerned, we are not so much of, we just have a few units there in case of our own IRA, in case of our, I think it is there also in Rajasthan, our centers like Kajri, they have a very good programs in the protected cultivation, and they are doing in the wonderful way. That, but only important thing here, we don't have a varieties in the protected cultivation. We are taking the varieties from the other country and the other hybrids from the other country and using our own materials. Using it is this. So you have to have our own materials by which we can also develop some of the programs in this particular aspect. So these are the new lines are coming up nowadays by the different public sectors organization. They put effort that we should have some of the varieties to be grown or can be grown in case of your protected cultivations, in case of tomato, in muskmelon, in cucumber. Even in case of bitter gourd, we have a variety, as you mentioned, Sarasda, which is also very much used for the protected cultivation. And now in case of bitter gourd, as you mentioned, because I am working in bitter gourd, I can say that we have a variety of Pusa Rasdar. By spraying with the GA without pollination, you can also get the fruit size about 100 to 100 grams of 80 to 100 grams of fruits. So that also work we already started here. I think this type of combinations of the chemicals by which you can also induce the parthenocarpic type of fruits in case of bitter gourd and any other crops. So that also will for the good use on the protected cultivations. We have to also consider this one. And then tremendous increase in the training system and other system by which you can also have the size of the fruits you can manipulate, number of fruits you can manipulate, whether it is tomato, in case of capsicum, the quality of the fruits can also be enhanced with respect to different training and other system. The grafting is very important nowadays. We have to, just to initiate it in the country. I think VNR Seeds done a lot of work in this particular grafting, but other than them, we are very, very limited in this grafting. I don't know, many of these our viewers may be knowing that India, we have to go for grafting, which is a very good potential. If you see in China, if you see in Brazil, in case of your Japan, you can see 80 to 90% they have the grafting crops in Solanacea vegetables as well as the cucurbitaceous vegetable. But we are just standardizing our rootstock, whether it is resistance to diseases, whether resistance to the abiotic stresses, particularly the high moisture level or the temperature conditions, that we are just practicing. And we have seen many of the materials now screened by the institute to have resistance to the abiotic stress, which is very, very important. And that can also be this one. This I put it as a fancy sake of this one because it is very, very important nowadays. People have to have a vegetables to be more, how we can propaganda that in a once vegetables plant you can grow 3 crop at a time. So it's very important because now this kitchen garden, because of the Poshan Suraksha vision, in the villages, in case of your schools, they have type of fancy type of things they require to have. So my concern here, by the simple grafting technology, you have a pots in your terrace. You can also grow vegetables, 3 vegetable crops in a one plant. So this is what I put it here. Not that it is so much of big things, but you can have a grafting technology where you can have your own ability to produce the crops as time. But now you see these problems in the country as a whole. We have a different type of pest management strategy we have already there. The organic pest management is very, very important nowadays. People are thinking about this natural farming or organic farming or the chemical-free farming is coming in one day. But as you mentioned that we are already discussing that natural farming cannot solve the purpose of this to feed the whole country as a whole. But at least some of the area we can go for that one, where there is a naturally people grow the vegetables without giving any inputs. That is the only part. Other things, the organic farming, which is very, very important. Now this government is also trying an organic farming, the mode of operandi, particularly for how to go for the organic farming. They are now having the guidelines they are creating. I think that sort of things also to come up. To here you can use the biopesticide. We are talking of the biopesticide very long way. We know that there's something like trichoderma, and all the things are already used by these people long back. But what type of this upscaling of these particular agents, or particularly the biopesticides, is not been done so far. So that also we have to go for, whether it is an NPV virus or there is an parasitic predators. We have to take it that this is a long way, particularly for the vegetable cultivation is concerned. So that part I already mentioned the some of the stories of this one. But what way again we have to go for in the next decade? We have to again, those are research we are, as I mentioned, many of these crop improvement programs still is going on, and we are very much of lag behind than that of the other countries is concerned. The people have tried the basic science research, they mixed with the applied one, and they have a lot of improvement already been done in the vegetable research. But here we have a lot of problems in our country, but still we are struggling to have the infrastructure. We are having to have a different other methodology to go for vegetable crop improvement program. If you see the challenges we have climate resilient crops to be developed because we have climate change. We have to have a productivity increase because also to see the input efficiency, use efficiency to be improved in our own materials. Those are to be go for the crop improvement program. Conservations of the resource and soil health, which is very, very important. Our soil becomes sick. So the nutrient efficiency, use efficiency, the use pesticide what you use, that's also we have to take care when you go for these are the breeding programs or the crop improvement program or the improvement programs in the vegetable crops as a whole. We have to see this quality of vegetables in the global market. If you see the export vegetables as a concern, definitely we are not in the standard with respect to the quality, with respect to the size, shape, color, everything is you have to go for very critically to develop this or to see in the global markets there are vegetables to be grown in to be exported to other countries. New diseases and pests because of the climate change and the development of resistance among different pests, which is very important, and post-harvest management value addition, which is still going to be a long way to go for this post-harvest management of vegetables crops is concerned. The productivity and the uneven productivity in the different states or different states is concerned. We have a state where we have 25 to 30 tons per hectare in case of Tamil Nadu. We have the states where the less than 10 tons per hectare in case of northeastern regions, particularly Nagaland, Mizoram, even Rajasthan, Sikkim, and Manipur. We have a very, very less productivity is concerned. So that gap we have to fill up so that we can definitely. These are the challenge. These are the opportunity where we can have also our own program to be initiated. The non-traditional or non-productive areas we have to go for vegetable production. If you see the global increase in the temperature, you can see there is already 100, 200 years or 100 years, we have increased about 1.2 degrees Celsius of temperature. And definitely by the process of increasing temperature, we are reducing there is reductions in the crop yield, crop quality, or vegetable production as well as the vegetable quality is concerned. So that part we have to take care when you go for a crop improvement program. Climate change, as you mentioned, we have to have a different people are coming in the malnourished way, and definitely for them we have to create some of this improvement program which can also have to reduce the or we can also tolerate to the global warming. The global warming or climate change, which leads to a, you can see in the map where you have only the problems is arising with this global whether it's a flood, whether it's a drought, whether it's a biodiversity loss, or whether it's any things or ozone things are there. Many things are there with respect to this one, and definitely we have a diversification. The land degradations are there. We have a stratospheric ozone depletions there. We have altered precipitation is there. So there are a lot of changes is occurring in this weather and the climate change, and that part we have to also consider when you go for global warming is concerned. If you see this changing the pest scenario in vegetables, some of the year this like in case of serpentine leaf miner, which is there only in the tomato. Now we can see presently infested in many of the solanacea crop. Even in the peas and beans, we have this particular crop, particular leaf miner is there. If you see the white flash, in case of guava and citrus, it is in Vendi. If your Hada beetle is one in brinjal, now it is in bitter gourd. So in this crop is also, this insect pest is also just invading the other crops is concerned. So these are some of the challenges which is very, very important to we have to address, because gall means was in brinjal, now it is chili and capsicum. You have red spider beetle okra now in case of brinjal, chili, in case of coffee and Indian beans. So that type of challenges also new pest scenario from one host to multiple hosts they are coming up. So that is in case of vegetables history going on. So we have to work upon that. Then we have a biotic stress. A lot of biotic stress. You see any crop in vegetables, in tomato, we have different type of biotic stress. There's a lot of multiple resistance varieties to be developed. These are some of the points I put it here because these are some of the important things we have to go for when you go for breeding or crop improvement program or any other programs by which we can manage this load of the pests and diseases is concerned in vegetables. So these are very important nowadays. And to see this loss is concerned in caused by major insect pests, you can say still we have a problem of diamond backmark, which is 50%, 2% in loss is there. We have chili thrips. Recently, we have in chili thrips also there in Andhra Pradesh and Telangana, where there is 100% loss. There are about 20,000 hectares of this particular chili thrips is was this put it there. So my point is that we have to very much of important with respect to the crops where we have pests or diseases. There is sufficient, there is a lot of losses we are also facing, and that we have to work upon this one either through this crop improvement program or through these management practices, which is very, very important. If you see, consider the disease also again is very important for us. Leaf curl viruses in tomato, you have early blight, wilt, nematode, everything is there. So these things are also to be considered when you go for breeding or improvement program in this particular crop. Many of the things we are also doing, but now, as I mentioned, that it is not the conventional way of breeding where we can also achieve our goal. We have to definitely go for next generation breeding program for the crop improvement, which is very, very important nowadays, which is very, very essential for us. Otherwise, it is not. We cannot cope up with these challenges. What I have described here, we cannot make the dent on particular for having this type of things. We can have the biotechnology approaches like rapid regeneration advancement. We have to have productions like double haploids, genomic selection, marker selection, backcross breeding, or whatever you have to have in the biotechnology approaches. We have to definitely increase the way of the process for this one. Now, this phenomenon of speed breeding, which is very important, as you mentioned, you cannot wait for the last 8 to 10 years to have this type of programs to be done it here. But at least we can also have to use the next generation breeding program, which must associated with this genomic assisted breeding, next generation sequencing, and pangenomics. So these are what we call as supra pangenome, where we can use our speed breeding program, particularly phenotyping of these traits, then association of the genes with respect to this particular phenotyping, through which we have to go for other methods also, like you have to identify the genes, clone the genes, go for speed breeding, go for genome editing, and also phenotyping, which is not in the way that what we are now doing. It's not the biparental mapping system. Still, we are doing in our program, which is not required nowadays. We have to go for modern approach by which we can also in a short period, we can go for this one. We have to have a sequence-based trait mapping approaches, whether it is QTL-seq, whether it is in the BSS system, whether it is you have GBS, whether you have a GWAS system, we definitely have a different modern operations with respect to the crop improvement program. We have to have a pre-breeding program because already our materials, what we have in the world, we adjusted. We don't have a specific resistance to biotic and diabetic stresses. We have a different accessions of the species are available, or the related species are available in this country. We have to use or introgress their genes in our own varieties by which we can have a resistance to different stress and biotic and diabetic stresses. These are we have to create. We have to see the genetic diversity of our own Indian materials. We have to go for phenotyping properly with respect to the trait is concerned. Also, we have to discover the haplotypes, alleles, and the new varieties of target, which is very, very important in our programs. That is also required, particularly for the vegetable crop improvement program. Marker state breeding is now, it is not new to all. Now it is, everybody knows the marker state selection. But how many of us were working in this particular aspect? How many breeders are working in this aspect? Very few, some of the labs they are working. But if you see in other countries, they already started this marker state breeding. And other crops, they really they have a tremendous achievements, but we don't have. But still, we have to again struggle more and more with respect to this particular aspect of this marker state breeding. Genomic assisted breeding is again a very important, what we call as 5 Z's. One is genome, then germplasm, genes, genomic breeding, and gene editing. So these things we have to combine together all. Without germplasm, we cannot do. Without genome analysis, we cannot do. Without the availability of genes, you cannot do. If you go for reverse breeding, or if you go for the reverse phenotyping or forward phenotyping genes identifications is very, very important. And then will also you can go for genomic assisted breeding and gene editing, which is coming up, the new way to go for this one. As you mentioned about this genomic assisted breeding, there are several techniques to use, like across inbred lines. You can use the magic population, you use the GWAS populations, where you can identify the genes responsible for the particular traits of interest. In that case, you have to have a very good way of breeding programs. You can see that some of the time, in the last part of this one, I put it here, that time setup is very important, where less time is consumed or short time is consumed, we go for that type of molecular assisted breeding. Particularly for the GWAS, it is very short time is required. The population structure, in case of other, it is not so good. So if you go for structured population, you have to go for GWAS, because in simple populations, you can identify number of traits, number of genes you can identify. But in case of bioparental mapping system, backcross mapping way, or magic, if you take it, is a very less amount of genes or traits and the genes you can identify. But if you have a GWAS, you can number of genes you can identify in a very, very short period. If you are now, as I mentioned, we have a tremendous size or increase in the way of our mapping population identifications of genes are there. Earlier, we used to have genotyping by sequencing. Now they have mapping by sequencing. You create a mutagenesis. Particularly in case of tomato, they've given an example that you have to create the EMS mutagenesis through which the color mutants you develop. You go for bioparental mapping system. Go for then this whole materials. You can use the whole genome sequencing methods by way of precise way of markers mapping. You can also identify the genes, the locations, and the SNPs associated with this one. So this is a very, very accurate way of mapping the genes or in the particular crop. Now I should not go for all the things, but if you see the brinjal as a whole, as a transgenic, I put it here. If you have a biotic stress, you take this type of genes and go for transgenic to develop the biotic stress resistant type of brinjal. If you have to insect nematode resistance genes, you can have a proteinase inhibitor, you have endotoxins synthesis genes. That type of things you can go for insect resistance. You have to have a quality, then you have to go for that is in case of gene editing by this SMPPO genes, or you can go for betaline synthesis by some genes, oxygen synthesis by some genes, and that also you can precisely do with this quality, particular for transgenic way of development of this one. Even to disease resistance also, some of the genes already identified, which you can also utilize this full way that the crop improvement or the genetic improvement on the vegetables can be done in a very short period. Now this is the way we have to go for trait discovery to trait genetic engineering. That is what we call as a gene editing. You started with the new traits, that is the phenotyping. We should have high throughput platform, a phenotyping platform, where you can have new diseases coming. So you have to go for the symptoms and all things to be analyzed very easily. Root architecture, particularly for the drought and moisture stress conditions, where you can have a different architecture of the roots that you have to study with a very, very systematic way. The way of which we can also use the different other methods like robotics. We can use the drone facilities for mapping, for phenotyping, which now is coming up very, very significantly. You have a screening genetic resources and QTL mapping. Still, we are struggling for that one, but we have to go for a huge populations, huge infrastructures to create ourselves to go for QTL mapping and reverse our analysis of these particular genes. Then marker selection, genomic edited gene editing, which I already described. Then also you can also see the gene and allele discovery by way of fine mapping, by way of allele diversity, RNA sequencing, or QTL-seq. You can go for that one. Now it's coming to this one, which is very important, is the genome editing. That already a lot of work has been done in other countries, but still we are struggling. If you see the genes already available with respect to the leaf and stem in case of tomato, if you see this meristem, if you see the roots architecture, biotic stress, the flower, the fruit size development, ripe fruits and ripening, and all the process, you have a different genes combinations have been identified. So you just target that particular gene, and you can definitely go for genome editing in this particular major crops is concerned. The people have tried, like CRISPR-Cas gene editing. In case of tomato, they have a different branching habit of the flowers, and the clustering pattern of the flowers can be changed with their 1 or 2 genes. You can go for CRISPR editing. You definitely can find it here. You can different way that the fruit clusters, you can find it. The shape and size of the fruits, they also can be increased or decreased, or you can have a change the shape of the fruits by the single gene, or we can just going for gene editing in a single that one nucleotide. These are the important way that people have already been done. So we have to concentrate, particularly for the viral resistance in case of different crops, whether it is tomato, whether it in case of cucurbits, you have a lot of viruses. But if you know some parts of that one, that which are the major genes is concerned to the resistance, definitely we have to target for this CRISPR-Cas also, and gene editing also, and definitely. And we also tried now, we have now in our lab is working on a YVM resistance in case of okra. We are also tomato leaf curl viruses gene editing we started. We also started going for the particularly for TSS, and that is processing trait gene editing in case of tomato. So this we have to already started. I think we already prepared some construct and all the things, and definitely will be succeed in the future with respect to gene editing is concerned. As I mentioned, the palm genomic study, which is very, very important to know that you have to have a unique material, so unique genes to identify through these palm genomic studies. We can see this cultivar 1, cultivar 2, and cultivar 3. So these are the different species or different varieties we have to go for genome sequencing. And from individual species or cultivar, we can identify the unique genes. And it is most now significantly done in case Solanum tuberosum, Lycopersicum, Capsicum, Enum, Raphanus, and Brassica. The people have a lot of work has been done to find out the unique genes with respect to the different trait is concerned. There is a different other tools like phenomic tools, as I mentioned. If you are not doing phenotypic properly, there is no way that you can go for any of this breeding methodology. So phenotyping should be precise. For that, we have a lot of other methodology has been done, lot of instrumentation already been created by the different people of this world, and they work on this particular aspect. And in the base itself, in the phenomic tools itself, the people can identify the good genotype. There is no need to have all the genotypes done in the field itself for evaluation. By the way of process and the way of phenomics tools, you can identify the variations or in the germplasm, the biodiversity present in your germplasm. Sorry, the genetic diversity present in your germplasms by phenomics. So that we have to consider. By the phenomic is also much more important than the genomics. By the way of phenomics, you can also identify many varieties, many lines you can develop, which is totally different. So that you can, by the simple observations, we cannot see it. By the visual observations, you cannot see it. But by the method of the tools using for these particular phenotypic evaluations, you can identify some of the varieties. So discovering mechanism of the genes are also important, because to know the traits, the genetics, which is very important, we have to go for reverse phenomic studies. That means based on the phenomics or genomic studies, you can go for phenomic studies. So this is a reverse phenomics, as well as forward phenomics. By the way of phenomic studies, you can develop the varieties and other hybrids in this particular crop. So these are the way we have to lot of things. So now I have only 2 slides are there. I think many things I described about this way of working, what type of things we need to have. I think this most important thing is that if you want to transform the vegetable productions of this country, we have to have some of the points to be remembered, some of the works to be carried out very precisely and in a very short time. We cannot linger the crops developing varieties for last 8, 9, 10 years. We have to evaluate them phenotypically precisely. We have to evaluate them and go for the study of the diverse of genetic resources. We have to go for developing the hybrids and varieties in that way. We have to go for the trait-specific genotypic identifications in that particular very short period. Now we have to consider also the quality, which is very, very important. We have to the concept of climate-smart breeding. We have to have genetic breeding population in the large number. Genomics mediated breeding we have to start upon. The molecular tools we have to use. High throughput phenotyping is very, very essential, and metadata management tools, big data analysis also to be done in vegetable crops. Otherwise, we cannot do the breeding actually what way we want to do. So that sort of things we have to go, and we have to definitely the biotechnological approaches or the techniques we have to also use for revolutionizing the process of worldwide agriculture system and the vegetable system is concerned. We have to also, as a complement program, not only this one, we have to also have the vegetable production system, which is very, very important. As I mentioned, like protected cultivation, grafting, then you have to have increase the efficiency, like in input efficiency, whether it is water, whether it is a nutrient, whether it is an insect pest management, that also we have to take up on. Then we have pollinators, which is very, very important, as I mentioned here, because many of the vegetables are cross-pollinated. If you are not looking for the pollinators, also you cannot develop. Whatever you develop is of no use if it is a cross-pollinated crop. So these are some of the things we have to go for. I think, thank you, sir. Thank you very much for the patient sharing. I think I took about, hello. Hello.
Any aspect of the vegetable you have not left,
Hello.
whether it's a production or improvement, whether use the genomics or phenomics or the futuristic one, everything you have covered within limited period of 1 hour or so. So excellent job done. Now I request Dr. Shailendra Rajanji, just highlight few points of his talk.
Thank you, sir.
Kindly don't cover the entire lecture because he had touched the entire gamut of the vegetable, not only the improvement but the production also.
Thank you, sir. It is very difficult to cover also everything.
Yes, yes.
But we should agree that it was an excellent, marvelous talk, linking us with all the aspects, not only the modern tools, everything was there, not from the production side, also vegetables improvement, everything. So I should say that is we have seen we had an overview where we are, how we are moving, what we have done, and how we have to perform. This is only for, and he has given a beautiful examples of all the aspects, particularly what the technologies are required for transforming the vegetable industry in the country. So I will not talk much. He has already given account of each and aspect in a summarized manner, and that too in a very, very simple language. I am thankful to Dr. Tushar Kant Behera sir for such an important and knowledgeful lecture given to us. Thank you very much.
Thank you, Dr. Rajan. Can I request Dr. Kalia to be with Dr. Tushar Behera, what he talked about? If he has missed something, kindly just
Yes, sir.
briefly explain.
Sir, as Dr. Shailendra Rajan has already said that we have had an excellent talk, exhaustive talk on all aspects of vegetable science, improvement, production, protection, whatever you talk. But I must say in the beginning, and the first and foremost point, that is the kind of result, the transformation in the right institute you see when you put a right person at right place. That is the first point, how you observe the thing, a person who has the capability of connecting people, extracting work from those who can be put at the right point, and also perceiving what is required for the future. Visionary approach and the connectivity amongst people is very important, and that you have seen in the entire talk. I need not to explain the talk, but the very important factor that we used to talk about when we were teaching students and talking in various programs, that in vegetable breeding, the important point from vegetables to make it saleable and get more price to the farmer and best quality to consumer is to breeder has to concentrate upon external traits. External traits breeding is very difficult, challenging, and a lot of opportunity, because that is the factor which Dr. Tushar Behra has touched. And I think uh, that point may be taken up in the seat of vegetable, which is now Varanasi IIVR, where all different aspects can be tackled in that sense, that external traits, gene identification, integration, and putting them into right track to bring out the product which can fetch a good price to the uh, farmers, and also the quality produce or requirement of the consumers is met. The tools and technologies elaborated which are available in the past, and how they became available during the process, and what is available elsewhere across the world in future, which vegetable breeders, scientists can concentrate upon to draw maximum possible benefits, is there. But for all this, we need to train our scientists who are not well exposed. Every day new technologies are coming, and IIVR Varanasi is such an institute which has different departments to bring out the right kind of technology for the end users. So the training program in each future technologies, the further upgradation of the labs they have is required, which should commensurate with the new technologies, which are expected technologies. Rather, they should also concentrate upon that they can also bring new technology, not that we should always look forward for new technology come and then we start working. We can also educate our scientists to work on such aspect that we bring new technologies which are useful for for our own crops, and the traits which our people look for, our consumers look for, which is very important. And so means that encompasses the challenges we have, a lot of challenges, the large number of crops and large number of factors which needs to be attended, which is important. But at the important is that what are important for us today, and what are in near future they need to be concentrated upon. And one important factor is that already we have technologies available, a lot of varieties available which are useful for the end users, that has been mentioned in detail by Dr. Behra. But I think we need to concentrate upon how to make them available to the end user, how to popularize them. That factor is lacking, and I think we had been discussing, and they have already been taking through AICRP, taking this factor that we have to compete. We have spent a lot of, the country has spent a lot of money on these development of these technologies, and if end users, the poorest of the poor, are not able to enjoy the benefits of these, then I don't think there is any benefit of these. So that aspect now needs to be also concentrated upon with the new uh, future improvements and the production aspects that we have to work upon. With that, I thank the speaker profusely for the nice presentation. I hope the viewers must have been educated and they must be benefited, especially the young scientists and the students must take imbibe in themselves that we have to do. We have to, I mean, uh, take up a challenge and the aspect they are interested, and concentrate upon there and contribute. With that, I thank you very much, Dr. Behra.
Thank you, thank you, thank you, thank you, Dr. Kalia,
Thank you,
for
sir.
adding to the what Dr. Rajan had said, and elaborate lecture by Dr. Behra. So, friends, we had wonderful, I think, talk by the topmost person on vegetable crops in position at Varanasi, the holy place. With the blessings of the Shiva, we further aspect that he will have new and newer technologies for boosting the vegetable production in the country. Not only the production, but the nutritive vegetable. Friends, I am one of the oldies, and we used to believe on the breeding methods and religiously used to develop the varieties, and developed varieties are lasting that we can claim. But I lay emphasis on that. Those days have gone. We have no patience. We want everything quick. Whether it is a development of some variety or something else, no patience. And luckily, the science has advanced to the extent that the quicker deliveries are there. The tools and technologies are there to deliver the uh, desired thing. So we should resort, my message is that we resort to the new technologies. Please keep the other things in a low key, what the people talk about. But if you want the improved thing, desired thing, quality things, things in time, and you want to retain the youth in the agriculture or vegetable production or whatever it is, we want the white collar farmers to produce the vegetable. I'm sorry to say that people may laugh on me, but the era is that we should have it, and only the technologies can help. Technology, Dr. Behra rightly included it in talk, though I was not expecting that he will include the production technology also. But rightly, now I realize, rightly he has included in that, because overall, whatever the crop improvement is there, the results come through the production only, and the production technologies do contribute to that, what are the so-called genetic potential or things like that. See, we have got the varied requirement, even within the country, what to talk about the entire world, and we have no patience, really. And really, why should we have the patience? We have the limited life span. Why should we have the patience? We should get the thing the quickly as quickly as possible. So modern technologies, but technological tools, what all here talked about the genomics and phenomics, all those technologies, and further the technologies are improving upon. We are reaching gradually to the nature, what is there in RNA, DNA, and the gene, all these editing and whatnot. And number of technologies are coming up, but all those are the what were happening through the nature. We are trying to understand them. The moment we understand, we are already we jump upon, and we want to quicken to develop these varieties. So what I want to convey, that respect the new technologies, and respect the institutions which are developing it, and the institutions should take up the modern technologies seriously, ignoring, I emphasize that, what the people will say. That you can see it, as in case of what you call that transgenic, I don't know, I don't want to use the word, some word I use for that, uh, transgenic one. There are alternatives to transgenics there. You don't bring the gene from the different species and different species into one. Have the intra-specific. Intra-specific is not intrageneric. Intrageneric will also do. Don't go to the animal kingdom and the plant kingdom and mixing them together. Have it vegetable genes in vegetables. Why there should be any objection to it? And country is rich in that. And I think the richest country in that vegetable, see, all what you talk about, they are available in the nature. Only thing we have to develop the tools and techniques to just possess them. They are there in the nature. So fantastic talk, and I'm thankful to Dr. Behra, despite his very busy schedule, he could prepare such a nice, exhaustive, and educative lecture. I'm sure the viewers must have been benefited by this, and we will be in touch, and may like to have his lecture in near, if not near future, sometime, as and when he is free on some other aspects. Thank you so much. Thank you, Dr. Behra. Thank
Thank you,
you,
sir.
Dr. Shalin, Dr. Kalra, and the manager of that studio,
Thank you,
the
sir.
person who is doing the job and making us to work, and making us to be useful Otherwise, it becomes difficult if something goes wrong, the internet and whatnot, whatever is there. So I'm thankful to one and all, and I think we will meet next Sunday with the topic which Behra has already introduced, the organic farming. So organic horticulture, we will have the detail this one. Till that, bye, and
Okay, sir. Good afternoon.
enjoy rest of the Sunday. Thank
Thank you,
you
sir.
very much. We meet
Thank you.
again.
Thank you,
Thank you,
sir.
Dr. Behra. Thanks a lot.
Namaskar,
Thanks a
sir.
lot.
Namaskar.
Thank you.
Namaskar.
Namaskar.
Thank you,
Namaskar.
sir. Thank you.
Thank you, sir.
Okay, ji. Thank you so much.